One of the main goals of synthetic biology is to create genetic parts that help scientists build genetic circuits. Genetic circuits are a collection of genes that work together to make a cell express a new ability, like detecting the presence of a toxin or performing logical computations. Synthetic biologists modify existing DNA by either adding, deleting, or on-the-spot editing of a genetic sequence. This new DNA can then be inserted into an organism, causing it to express new proteins and thus giving the organism a new function. For example, a synthetic biologist could take a gene for green fluorescent protein from a bioluminescent jellyfish and insert it into an E. coli to cause it to glow green. A lot of progress has been made in the ability to control genetic circuits for properties like gene expression strength, where well-defined ribosome binding sites can be swapped with other ribosome binding sites to translate more of a protein. A ribosome binding site is the sequence where, once DNA has been transcribed into messenger RNA, a ribosome will bind to begin translation. It is this sequence of base pairs which makes the ribosome more or less likely to bind to the messenger RNA and therefore makes it more or less likely for the gene to be translated into a protein. However, controlling gene expression speed has been more difficult than controlling gene expression strength. You can't control gene expression speed in a predictable way by simply changing the sequence of some genetic part. Past approaches to changing speed aren't ideal for synthetic biologists who want to tune their circuits because they require major changes to the existing circuit and results can be unpredictable. Our team aims to design a way to control the speed of gene expression, where a pre-characterized genetic part can be inserted into the gene to tune its expression speed in a predictable and simple way. Mathematical analysis shows that we can reach steady state, or the point at which there is a consistent amount of protein in a cell, more quickly by increasing the rate of protein degradation. However, we could find no thorough experimentation of this relationship in the literature, making this an ideal subject of research. Our approach to accomplish this goal is to build a collection of parts which can be swapped into a genetic circuit to modify a protein's degradation rate. We will accomplish this by using protein degradation tags. A protein degradation tag is an amino acid sequence that is attached to the end of a protein and signals to a protease to destroy that protein. Our collection of protein degradation tags will allow bacteria to target a specific protein for degradation, using a well-defined protease that recognizes the protein degradation tag and destroys the associated protein. This system is effective because each tag has a different rate of degradation, allowing us to measure if a faster degradation rate allows for the genetic circuit to reach steady state faster, which can increase the speed of the entire circuit. By characterizing and controlling gene expression speed using degradation tags, our team will provide new results and parts that can modify existing genetic circuits, an important tool for iGEM and the field of synthetic biology as a whole.